Welcome back. This is Shay. I'm recording this video on January 19th, 2023. This should be the Thursday before your lecture on Monday. We left off with functional groups last time. And again, if you remember what I said in the previous lecture preview, I was just letting you know that functional groups are important. They are going to determine what molecule that you are dealing with. For example, amino acids will be amino acids because they have an amino group, which is just a nitrogen bound to two hydrogens. For our purposes, I just want you to be familiar with these functional groups and their terms. They will be important for those of you wanting to go on to professional school. You'll memorize them as you go on. But for our purposes, I just want you to be familiar with these groups. You don't have to memorize them, but I want you to know whenever I talk about a hydroxyl what I'm referring to. So just to show you exactly what a functional group is, a functional group is going to be a group that is attached to a carbon, and it's going to give the molecule its properties. So a hydroxyl group is just an oxygen bound to a hydrogen. We call a carbon double bonded to an oxygen a carbonyl group, as you can see here on the left-hand side of the screen. This C double bonded to an O, this is the same thing as a carbonyl, right? But it just has a hydroxyl group attached, right? This is called a carboxyl group. So again, once you have a carbonyl group bound to a hydroxyl group, we call that a carboxyl group. An amino, we just previously talked about that. This is going to give amino acids their property by having an NH2 bound to it, and then a sulfhydryl group. So just be familiar with these. Again, I'm not going to be really picky and make you guys recall these, but again, for those of you going to professional school, I think it is important to just have some familiarity with these at this point. What is important is the next group that we're going to talk about, the carbohydrates. We're going to get into the organic molecules. The organic molecules consist, if you remember, they consist of carbohydrates, lipids, proteins, nucleic acids. Organic molecules are going to be things that consist mostly of carbon and hydrogen. When we talk about carbohydrates, I want you to know that they are going to provide most of the energy of the cell. They're built from simple sugars, so you have something called monosaccharides, meaning one sugar. Saccharide just means sugar. And then disaccharides, two sugars. What is unique about these carbohydrates are the way they are assembled. This is always an examination question, talking about the ratio of carbon, hydrogen, and oxygen. They are in a 1 to 2 to 1 ratio. They're going to serve as primarily the source for generating ATP. If you guys can remember from Bio 112, we talked about the ingestions of carbohydrates. So the ingestion of sugar is broken down by amylase, a digestive enzyme. Once we get on an organelle level, we talk about glycolysis occurring in the cytoplasm and generating ATP. So again, just going to show you that these sugars are going to provide the chemical energy for generating ATP. And if you remember from the beginning of the lecture, what did we say chemical energy was? This was just energy that was associated with compounds and molecules. So the sugar or the carbohydrate has a form of chemical energy. This is potential energy in the form of carbohydrates. Once it is metabolized or broken down, it can then generate ATP, which is used then for movement and can be transferred into kinetic energy. So again, the important things, knowing carbohydrates are sugars. They provide much of the energy of the cell. They exist in a 1 to 2 to 1 ratio with carbon, hydrogen, and oxygen. I think knowing that glycogen is stored glucose, that's important. Knowing that glycogen is an example of a carbohydrate. Monosaccharides and disaccharides. Again, this is getting picky, but monosaccharides and glucose, fructose, and galactose, those are all mono and disaccharides. Those are the simplest sugars. And then from there on, We can start adding glucose monomers together to make glycogen. That's polysaccharides or mini sugars. And then cellulose. And if you remember, we talked about cellulose having that alpha 1,4 glycosidic linkage in bio 112. We said that we couldn't break down alpha 1,4 glycosidic linkages. That's why it was used as fiber. So lipids are going to primarily be used for energy supply as well as building cell parts. Lipids are made of different molecules such as lipoproteins and fatty acids. Lipoproteins are going to be exactly like the name says. This is going to be both a protein and a lipid portion. There's going to be many lipoproteins present within the blood plasma. Then knowing the difference between saturated and unsaturated fats. Saturated are solid at room temperature. They contain only one single covalent bond versus unsaturated fatty acids, which contains one or more double covalent bonds. This may provide a kink, therefore they're liquid at room temperature. So what's important with lipids? Knowing that they also supply energy and are used for building cell parts. Do you guys know what organelle lipids are primarily responsible for making up? That's right, the plasma membrane, right? Remember the phospholipid bilayer, the two layers of the phospholipid. So saturated, S for solid, unsaturated is liquid at room temperature. Here's an example of some lipids here. You have phospholipid. This is the hydrophilic head, which contains the phosphate group, meaning the fact that it's hydrophilic. Phil means an affinity for hydro, so it has an affinity for water. Therefore, it's going to be polar. Then you have the nonpolar fatty acid tail. So the fatty acid tail is going to be the nonpolar portion. The saturated, you can see that it's saturated. There's no double bonds here, whereas the unsaturated has a double bond, like we previously just said. This is just a picture showing lipoproteins. This is an example right here of a lipoprotein. You can see the lipids attached to a protein monomer here. Apolipoprotein B100, this is going to be responsible for cholesterol transport within the blood. I guess we'll get into that into biochemistry. Here is just another picture depicting saturated and unsaturated fatty acids. Again, we said primarily the difference between saturated and unsaturated fats is that saturated contains only single covalent bonds, whereas unsaturated will contain a double bond. And we know that saturated is solid at room temperature, whereas unsaturated, because of this double bond, it forms a kink and therefore is going to be liquid at room temperature. This is considered a carbon skeleton, like I was previously just talking about. As you can see here, the different carbons bound to each other. The functional group here, does anybody know what this functional group would be? This would be considered a carboxyl group. See, you've got a C double bonded to an OH, a carbonyl, bound to a hydroxyl group. So we would call that carboxyl. Moreover, about lipids, we have different forms of lipids. We have lipoproteins, fatty acids. We have something called triglycerides. And triglycerides are going to consist of 3 fatty acids and a single glycerol molecule. And essentially that makes sense if you think about the name triglyceride, right? 3 and then a glycerol molecule, tri, meaning 3 fatty acids and then a single glycerol molecule. Triglycerides can exist as oils or fats. Fats are going to be mostly saturated. If you think about cooking lard, it's going to be more solid, whereas the unsaturated fats are going to be more unsaturated. So whenever you're picking which to cook with, your things like olive oil, coconut oil, things like that, which are unsaturated, are going to be actually a little bit better for you. Steroids are going to be an example of lipids. Again, these have 4 hydrocarbon rings. This is why I pose the question, would it be beneficial for um, humans to have zero cholesterol. If you went to your doctor and he said to reduce your cholesterol, would it be desirable, if it's even possible, to get it to zero? The answer to that question would be no, it wouldn't, right? Because why? Lipids, such as formation of cholesterol, right, is going to be important for the formation of things like steroids. And your body needs steroids in the forms of androgens and estrogens, as well as cortisol and things of that sort, in order to function at a hormonal level. Excuse me. Lipids are insoluble in polar solvents such as water, and thus are hydrophobic, and we know that because of the fatty acid tail. So again, the main points for lipids that I primarily want you to know the different types. Lipoproteins are just a lipid plus a protein. They're present in blood plasma. Triglycerides, I want you to know that it's a three fatty acids bound to a single glycerol molecule. Know the difference between saturated and unsaturated fat. Saturated, primarily solid at room temperature, whereas unsaturated is mostly going to be your oils, which are liquid. Know that phospholipids are going to have a hydrophilic head, which contains a phosphate group, and a hydrophobic tail, which contains the fatty acid group. And know that phospholipids, or lipids in general, are going to be responsible for steroid synthesis. Steroids having a four-ring carbon molecules, hydrocarbons. This is just more examples. Here is the triglyceride that we previously just talked about. Here's your glycerol molecule attached to your three fatty acid tails. This is an example of glycerol. Again, tri meaning three fatty acids. Right, you've got the 1 to 2 to 1 ratio of your carbon to hydrogen. Again, with a glycerol molecule here. This is just showing you the difference between the hydrophilic group containing the phosphate. The negative charge here makes it polar. This is considered an ester linkage. You don't have to know that for our purposes at this point. This is the hydrophobic fatty acid tail. It's important to know that steroids are derived from cholesterol. Cholesterol is an example of a lipid. And if you previously remember on the previous slide, I said that steroids were 4 hydrocarbon rings. There's 1, 2, 3, 4. Okay, so 4 hydrocarbon ring. These are examples of steroids. So if you went to the doctor, you had pneumonia, and they decide to give you a shot of dexamethasone, or they give you a shot, right, a steroid shot. This is the hormone that they're giving you. It's a cortisone shot. Because my 139 class, we're talking about the function of cortisol and how it has anti-inflammatory properties. So this is the hydrocarbon, 4 hydrocarbon rings, showing that it's a steroid. If you remember, steroids are derived from lipids. This is testosterone. This is an androgen, right, that's responsible for puberty within males. Gives the deepening of the voice, the facial hair, the acne, the increase in growth height and muscle mass. It's estradiol, female hormone. That's also needed for development within the females. Cortisol, anti-inflammatory properties as well. This is a picture just depicting two bodybuilders who are obviously on anabolic steroids, right? Steroids meaning derived from cholesterol. These are anabolic, so what type of reaction would this be considered? What type of reactions would be occurring? Synthesis reactions, right? They're building, they're taking amino acids and they're building proteins, building polymers from monomers. This is something called an eicosanoid or icosanoid. Icosanoids are essentially just prostaglandins, and this is an example of an icosanoid. Icosanoids also use lipids. Here is a picture of the fatty acids that are used. Icosanoids are going to be important for inflammatory processes. So let's say that you have a headache, right? And you have a headache that occurs. Let's say you've got some tension in your shoulders because of the upcoming exam, and you decide to take ibuprofen. Well, the eicosanoids are going to be prostaglandins, which produce pain. So your body has this inflammatory process, prostaglandin E2 for fever and pain. So it's going to be responsible for causing a headache and stimulating the pain response. We can give a medication called ibuprofen, which inhibits the enzyme cyclooxygenase, which occurs right here. Arachidonic acid. This is going to be a part of a metabolic process within your body, and there's an enzyme here, cyclooxygenase, that produces the prostaglandins. What's the point of this picture? Just to show you that eicosanoids have lipid properties. So lipids are important for everything. They are important for lipoproteins, proteins present within the plasma. It's responsible for cholesterol transport. It's responsible for the formation of steroids within your body, such as androgens, estrogens, cortisol, and also helps with inflammatory process by formation of prostaglandins. So just kind of throwing that out there to you guys. Switching over from lipids, we're going to get into proteins. Proteins are going to be primarily, they're going to comprise 12 to 18% of the normal lean body mass. There are things called monomers, which are mono means one. So one um, amino acid, there's 20 different monomers of amino acids, 20 different amino acids, and how those amino acids are combined together will determine what type of protein is formed. Once we go from monomers and get into polymers, we're then going to talk about the formation of a dipeptide. And this is just two amino acids linked together. And I have in here talking to you guys about the central dogma. And do you remember how we formed these amino acids? We formed these amino acids through transcription, right? Transcription turned this DNA into messenger RNA and then eventually got it into RNA. And from there, we can combine these nucleotides together, and three nucleotides together is going to form a codon. And from there, translation is going to occur. And then you're going to have protein synthesis. So the combination of these amino acids is going to form the protein. And this is why I said that amino acids are the building blocks of proteins. Because again, during this translation process, we're actually going to begin to combine these amino acids to form dipeptides, tripeptides, and polypeptides. As the name indicates, dipeptide means two, tripeptide means three, and then poly meaning more than one peptide. What is important on this slide, besides knowing that amino acids are the building blocks of proteins, is knowing that they're connected by a peptide bond. And it's also important to know the structure of these proteins. How are they organized? I gave you guys a great way to know these last semester. If you were in my 112 class, again, I'll show that for this class. Primary structure is going to be just a linear amino acid sequence. This is just going to be your most simplistic organizational unit. Whenever you hear about secondary, I want you to think about alpha helices and beta pleated sheets. Alpha helix and beta pleated sheets are going to introduce turns and kinks within the protein itself, which is a buzzword that they like to throw out on exams. You may see it on my exam in the future. If I said that a scientist was studying a protein and he noticed that it was in the had a conformation of a 1 alpha, or had an alpha 1,4 glycosidic linkage, and it formed an alpha helices because of this. Which of the following protein structures was this most likely? Primary, secondary, tertiary, or quaternary? Because I give you the buzzword alpha helix, right? Because the alpha helix was forming, this was a secondary protein structure. So that's a way I could ask that in the future. If I were to say that it was in a three-dimensional shape, what would that be? It's tertiary, absolutely. And then quaternary is just whenever there's more than one polypeptide chain associated. So a good example of this is hemoglobin. You have 4 heme molecules bound to a globin chain. This is an example of a quaternary structure. So again, just to go through that, it's important to know that proteins are the basic building blocks are amino acids, and that amino acids combine through peptide bonds in order to form proteins, dis two, tris three, and then polys more than three. Knowing the different organizational structures, primary being a linear sequence, secondary being the formation of alpha helices and beta pleated sheets, tertiary being three for tertiary, right? 3D shape, and then four is an association with more than one polypeptide. Moreover, on proteins, again, like I previously talked about when we were talking about amino groups being a functional group, the amino acid sequence itself is what's going to determine the protein conformation. It's how they combine with each other. And then we know that the protein conformation will determine its function. Okay, so structure determines function. That's a very important um, concept to become familiar with, right? So, uh, if I said exposure to excessive heat, that's an important one, right? Radiation, electricity, or certain chemicals can denature proteins. What does the word denature mean? Well, denature means to change the structure or to unfold the protein. So I pose the question, why is it important to lower the body temperature when patients have a temperature of 104 or 105, especially for a prolonged period of time? And it's because of this denaturation process, right? When our body is subject to heat that these proteins are not don't function the best in, right? They don't have the best function in this excessive heat. They can undergo denaturation. It can alter the shape of the protein, and like I just told you, if you alter the shape of the protein, you alter the structure, right? Which means you're going to alter the function as well. This is why it can lead to things like seizures, um, maybe even death if it's not quickly corrected. Here are the 20 different amino acids. Again, if you're taking organic chemistry, this will be important to know. But for our purposes, I just wanted to show you that it is, we can go over the basic structure. You have a carbon group bound to this uh, carbonyl group, this C double bonded to this O up here. This NH2 is an amino group. And what is attached to the side chain, that is going to determine the functionality of this amino acid. This side chain determines the amino acid, which one it is. This is a picture just depicting to you what protein structure is. I think it's a very good picture, one that would be very useful for studying purposes, one you may see on an exam or on a quiz. This here is a primary protein structure. This is just a sequence of the chains of amino acids. It's linear, there's no folding. As you can see here, we have alpha helices and what we call beta pleated sheets. This occurs when the sequence of amino acids are linked by hydrogen bonds. So this is considered an alpha helix. This is a beta pleated sheet. Once we have more folding on each other, we can then begin forming a 3D shape. Again, you're still going to have the alpha helix here and the beta pleated sheet as well, but it's going to form a 3D model. Quaternary is just going to be association with more than one polypeptide. You can see here by the darker gray and the lighter gray shaded area, there's more than one amino acid chain present. Continuing on with proteins, enzymes are a type of protein, right? Enzymes are a type of protein, and they're responsible for catalyzing or speeding up metabolism. So they're a catalyst or they speed up metabolism. There's two different portions of enzymes that I think is important, the apoenzyme as well as the cofactor. The apoenzyme is the protein portion, whereas the cofactor is the non-protein portion. Again, guys, this is just simple memorization. So, proteins are an example of enzymes. Enzymes are responsible for speeding up reactions. Enzymes are made up of two different things: the apoenzyme, which is the protein portion, and then the non protein portion, which is the cofactor. Vitamins are going to be examples of non protein portions known as cofactors. Vitamin B12 is a very important one, vitamin B6. These are all needed for these different metabolic reactions to occur. This is also very important that I've discussed earlier on in this lecture as well as in class. Enzymes use substrates in their active site, right? Each substrate is specific for a particular enzyme. I use the example of amylase breaking down carbohydrates or sugar within the mouth. You have salivary glands which secrete amylase. This amylase is then going to break down sugars because they're specific for it, right? They're specific for this enzyme, or for this substrate itself. So once the enzyme actually binds to the active site's the portion on the substrate where the enzyme likes to bind. It forms something called an enzyme substrate temporary complex, and that makes sense. Think about the name enzyme substrate complex. And once the enzyme produces the desired effect, aka the products, the enzyme then just releases from the active site and then it can be reused over again. So, what's important from this slide? Enzymes. Enzymes are proteins. They speed up reactions. Consist of two different things: apoenzymes and cofactors. Apoenzyme is going to be the protein portion because it has the enzyme in its name, and then cofactor is the non-protein portion. I know that enzymes are specific for substrates. I know that they speed up reactions. I know they're reused, and I know that they bind at the active site. This is a picture here of what an apoenzyme and a cofactor may look like. Um, this is an apoenzyme, the protein portion. This is the cofactor, knowing that the apoenzyme, when it's present with the cofactor, forms something called a haloenzyme. Don't have to know that, but the substrate can then it can then bind at the active site. Nucleic acids. This is an important topic. Nucleic acids are going to be responsible for the formation of nucleotides, right? They're going to eventually constitute genes. We know that genes are the instructions that control cell activities and direct protein synthesis. There's two types of nucleic acids. We're going to have DNA and RNA. DNA is deoxyribonucleic acid, whereas RNA is ribonucleic acid. We know the difference between DNA and RNA is that DNA is double-stranded, RNA is single-stranded. DNA uses thymine, whereas RNA uses uracil. Um, Getting a little more specific, we know that purines are going to be double-ringed, and they're going to consist of adenine and guanine. And I just think of, I don't know why, but I made this mnemonic up, AGOG, AGOG, um, AGOG, or a dog eats Purina food, or cut the pie. CUT, cytosine, uracil, and thymine, cut the pie, pyrimidine. Just whichever you want to. If you think my pseudonym mnemonics are good, great. If not, then I'm sorry. So just knowing the base pairing for DNA synthesis, this is Chargaff's rule, right? A equals T, C equals G. Knowing that if I were to give you the amount of thymine, could you tell me how much adenine is present? Absolutely, because thymine binds to adenine, right, in DNA synthesis. We know that nucleic acids consist of three different portions. I've drilled this into your all's head: a nitrogenous base, a deoxyribose sugar, and then a phosphate group. Um, the nitrogenous base, again, is just adenine, cytosine, guanine, thymine, or if we're talking about RNA, that's uracil. We know that nucleic acids are the building blocks of nucleotides, right? Amino acids are the building blocks of proteins. This is, we're getting towards the end, ATP. Let's break it down to its actual name, adenosine triphosphate. So there's three phosphates present there, and then we have adenosine present. ATP, we know it's going to be the energy currency of the cell. Where's the majority of the ATP produced within the body? The mitochondria. Absolutely. It's the powerhouse of the cell. It's because all the ATP is generated there in the electron transport chain and the citric acid cycle, the majority being in the electron transport chain. There's an anaerobic phase and an aerobic phase. Anaerobic means can work without oxygen, and this is through the breakdown of pyruvate and lactic acid buildup. Muscle soreness after prolonged exercise. Eventually, the muscles become anoxic or not receiving enough oxygen present, and they wind up building up lactic acid, right? We don't have enough um, ATP for this to occur. There's a whole process that we can discuss in biochemistry that breaks this down. Aerobic phase, glucose is broken down, and mitochondria drive ATP synthase via hydrogen pump. So this is your electron transport chain and your citric acid cycle, or your Krebs cycle, whichever you're familiar with. Once used, ATP becomes ADP. It makes sense because we generate all of this energy through the breaking of this third phosphate bond. So ATP stands for adenosine triphosphate. It's this third phosphate bond that is broken that is responsible for providing all of this energy. Once that phosphate bond is broken, it becomes ADP, and then later it gets phosphorylated again. This is a picture showing what ATP kind of looks like. This is adenosine. This is just a simplistic version, right? With these two phosphate residues, right? Energy from glucose phosphorylates it, and then we get ATP. Once this phosphate bond is broken, we'll get ADP again, right? So ATP consists of three phosphates. We know it from the name: a ribose group and adenine. This is just another picture kind of depicting to you about this ribose group bound to this adenine, bound to these three phosphates. Through the energy from sunlight or from food, once this inorganic phosphate is broken, we then have ADP again. Um, so, again, guys, what's important for ATP? Knowing that ATP is going to provide most of the energy currency of the cells. Breaking of that third phosphate bond is responsible for providing much of the energy. Once ATP is broken down, it becomes ADP and is then later phosphorylated again to become ATP. Know that ATP is made of an adenine group, a ribose, and then three phosphates. I really don't have anything else for you guys. If you have any questions, please feel free to reach out to me. I hope this was helpful. Again, this is just a lecture preview. Watch it before coming to class. If you have any questions, you can go back to this lecture and revisit it. Again, if you have anything else, just reach out to me. Thanks.